Hello, everyone that's joining. We'll give it a few more minutes for some folks to join us. Okay, there's still a few more rolling in, so we'll start in about a minute. Okay, welcome everyone. We can probably get started on some housekeeping, and some more folks might roll in as we get going. So hi everyone, and welcome back to our Ask an Expert webinar series for the 2025 fall season. This is our third session in this series. My name is Meg Mills. I'm the International Watershed Coordinator with the Lake of the Woods Water Sustainability Foundation, and I also work part-time with the International Joint Commission, connecting with many partners across the Rainy Lake of the Woods Basin. I'll be your host again today and throughout this season's lineup of sessions. I'm also joined today by Tika Newton, the Foundation's Executive Director, who will help with moderating some of the question and answers at the end of this session. I'm joining everyone from Treaty 1 territory in Winnipeg today. It's pretty cloudy outside again, but it's a cozy day and a good time to learn more about our freshwater systems. Today, we're joined by Dr. Karen Kidd, Dr. Eden Hataley, and Dr. Rebecca Rooney, who will be sharing their insights on microplastics in the Great Lakes, a topic that's becoming increasingly important for all of us working in freshwater systems. The team's work comes from an International Joint Commission Science Advisory Board project called Monitoring Ecological Risk Assessment and Management of Microplastics in the Laurentian Great Lakes. Through this project, they've pulled together the latest science, built a framework for monitoring microplastics, and explored how to assess their ecological risks in a more coordinated way. And while the focus is on the Great Lakes, there are definitely connections to our own region. Here in the Lake of the Woods watershed, microplastics are an emerging issue too, as we know, and the approaches shared today can help guide how we monitor, assess, and manage them. So I'll just go over some Zoom basics. This session is being recorded, and it will be available to watch later at our website, along with our previous episodes. If you're new to the series, welcome. Ask an Expert is all about sharing the science and stewardship happening across Rainy Lake of the Woods water basin and beyond. To keep things running smoothly during the webinar, everyone will be muted and cameras will also stay off. When we get to the discussion part, you can raise your hand if you'd like to ask your questions directly. I can unmute you. You might need to unmute yourself as well, but you'll have the option to do that. Or you could type your questions anytime into the Q&A box. We'll keep an eye on that and circle back to them at the end. If I don't hear from you when your question comes up, I can read it on your behalf. Just a friendly reminder that Zoom also saves a record of all chat messages, including the private ones. So I'll be able to see those at the end. We'll wrap up today's session with some time for questions and reflection, so I really encourage anyone to join in on that. So just to introduce the presenters, today we're joined by three researchers. Karen Kidd is the Jaroslawski Chair in Environment and Health and professor in the Department of Biology at McMaster University. She moved to McMaster in 2017 after 14 years at the University of New Brunswick as the Canada Research Chair in Chemical Contamination of Food Webs. Karen is an ecotoxicologist, and her group studies the effects of forestry, agriculture, aquaculture, and municipal wastewaters on aquatic ecosystems and the accumulation of contaminants in freshwater food webs. More information about Karen can be found at www.karenkidlab.com. Eden Hataley recently completed a PhD in environmental science at the University of Toronto, where her research used a transdisciplinary approach to advance understanding of plastic pollution management in the Laurentian Great Lakes. Prior to her doctoral studies, Eden earned a master's degree in environmental studies and a bachelor degree in environmental chemistry, both from Queen's University. She is passionate about transboundary water governance and remains committed to working with diverse stakeholders to address complex environmental challenges. Rebecca Rooney carries out research in wetland ecology and is a world expert on biomonitoring and wetland assessment. Rebecca examines how human-caused and natural ecological disturbances influence wetland communities, including birds, invertebrates, and plants. She tackles fundamental questions around how communities assemble and what defines them, including the relative importance of biological interactions, environmental conditions, and landscape factors. She is keenly interested in the role of rare species and novelty in ecological processes. So with that, I'll hand it off to you three to begin the webinar.
Thanks, Megan. I'm going to share my screen here.
Great.
And thank you everyone for joining us for the webinar. I guess in addition to the bio that Megan read out, I'm also the Canadian co-chair of the International Joint Commission's Science Advisory Board and their Research Coordination Committee. And I'm joining you today from Waterloo, Ontario, where the Canadian geese are lingering on their southward migration and the fall leaves are starting to wrap up their autumnal display. So the International Joint Commission, or as we call it, the IJC, was created through the 1909 Boundary Waters Treaty between Canada and the United States. The IJC serves as an independent and objective advisor that assists in preventing and resolving disputes over shared waterways. The Commission also assists Canada and the US through the Great Lakes Water Quality Agreement, and we work to cooperatively restore, protect, and enhance the water quality of the Great Lakes. The IJC's roles under the agreement include providing advice and engaging the public. Under the 2012 Great Lakes Water Quality Agreement, 2 advisory boards were created to provide the IJC with expert advice. The Water Quality Board is the principal advisor to the IJC on matters of policy, whereas the Science Advisory Board advises the Water Quality Board and the IJC on matters of research and science. So the Science Advisory Board has 2 committees: the Research Coordinating Committee, which I co-chair, and the Science Priorities Committee, which my work group co-chair, Dr. Karen Kidd, sits on. And so that's a little IJC 101 to kind of situate where this report is coming from, because today we plan to discuss the findings and recommendations of a Science Advisory Board report on microplastics in the Great Lakes and how to strengthen efforts on monitoring, assessing, and managing plastic pollution. The formal report that we're talking about today is available on the Science Advisory Board's webpage, and this QR code will take you there, but don't try to snap photos of it now because we'll put it up again at the end of the presentation. So the Science Advisory Board's microplastic project has benefited from the expertise and guidance of its work group. Members of the work group come from across academia and federal, state, and provincial government agencies. This project also benefited greatly from the knowledge shared by workshop participants and guidance from the Chesapeake Bay Plastic Pollution Action Team, as well as the Southern California Coastal Water Research Project and the California State Water Resources Control Board. You'll hear from 3 of the 4 work group co-chairs today, myself, Dr. Karen Kidd, as well as Dr. Eden Hataley. But we have to acknowledge the important contributions of Dr. Chelsea Rockman, our 4th co-chair, who actually presented in this webinar series last week on her microplastic research. So a little bit of history. The IJC and its expert advisory boards have really been focused on Great Lakes microplastics for a decade. Our latest report builds on previous work by the IJC and other organizations. The first peer-reviewed study to measure and report on microplastic pollution in the Great Lakes was published in 2011. And the IJC Great Lakes Water Quality Board has maintained a microplastics watching brief since 2015. This watching brief summarizes information related to Great Lakes microplastics and plastics more broadly, spanning science, policy, and media. In 2016, the IJC hosted a workshop to develop recommendations to address the challenges posed by microplastics in the Great Lakes. Based on that workshop, the IJC published a report in 2017 that outlined 10 recommendations for Canadian and US governments to reduce and to monitor microplastic pollution. Significant work in the last decade has focused on the science of environmental and ecological impacts of microplastics in freshwater habitats. In 2018, the California legislature passed 2 bills that required the creation of tools to collect environmental samples for microplastic-related analysis, to quantify and characterize microplastics in environmental samples, and to assess and manage the risk of microplastics exposure. So I'd like to bring focus specifically to work by the Southern California Coastal Water Research Project Authority, or as we affectionately call them, SQURP, which is how you say that acronym. Since the bills in California were passed, SQURP has convened several workshops and work groups to develop these tools, and our work group was able to adapt those tools to the Great Lakes. So you'll hear more about that shortly. I should also mention that many of the people involved in SQURP's work were also involved in our IJC Science Advisory Board work group. Anyway, this all led to the creation of the IJC Science Advisory Board's Microplastic Monitoring and Risk Assessment work group. And the outcome of that work group is the subject of our presentation today. Our work group had 3 objectives. First, we wanted to synthesize recent advances and knowledge on Great Lakes microplastic science that had been published or was in process since that 2016 workshop. We also wanted to develop a framework for monitoring microplastics that would enable harmonized monitoring and reporting about their concentrations in the environment. And finally, we wanted to develop a coordinated ecological risk assessment and management framework that would support better assessment of the risks of microplastics exposure in the Great Lakes Basin. To meet our first objective on synthesizing recent science advances, we undertook a session on plastic pollution at the International Association of Great Lakes Research's annual conference in Toronto in 2023. And we also developed a literature review, which includes sections on the physical and chemical characteristics of microplastics, as well as a summary of field and lab methods, and a summary of environmental occurrence data for the Great Lakes, as well as toxicity data for freshwater species and regional policy and management considerations. We also uploaded that data into 2 publicly accessible databases. The first as the monitoring database that the QR code would take you to, hosted by the University of Toronto, where all of the data we were able to synthesize about exposure in the Great Lakes is housed. And the second database, called TOMEX, or Toxicity of Microplastics Explorer 2.0, should be available at the QR code on the right, and that would take you to the synthesis of ecotoxicological literature on the effects of microplastics for freshwater and marine species. This project reported on the presence of microplastics in water, sediment, and living organisms, which we call biota, and on shorelines in the Great Lakes and their connecting waters and tributaries. In this figure, it shows the concentrations of microplastics particles reported in surface water across the studies from our literature review. The concentrations per cubic meter range from less than 10 particles to approximately 10,000 particles in offshore waters and upwards of 100,000 particles in certain tributaries. Microplastics have also been found to contaminate algae, to occur in fish, birds, amphibians, and invertebrates. Generally, the most populated basins, like Michigan and Ontario, had the highest concentrations of microplastics in the water. Additionally, tributaries generally had higher concentrations compared to open waters. So I'll turn things over now to my work group co-chair, Dr. Eden Hataley.
So I am going to just briefly highlight a few studies that have been conducted in the Great Lakes region that examine potential human exposure to microplastics through Great Lakes resources. There are currently a very few studies that specifically focus on this area for the Great Lakes. And the first of those is a 2018 study published by Kosif and colleagues, and you can access that study through the QR code on the screen. In this work, researchers analyzed tap water samples from 14 different countries, and including the United States. And in the United States, this data set included 8 cities across the Great Lakes region, and you can see them mapped out here on the slide. And across all of the US samples, tap water contained an average of about 9 microplastics per liter, with concentrations that ranged from 0 up to 61. And just for comparison against the global average across all of the all of the countries that were looked at in the study, that was around 5 microplastics per liter. Next slide, please. In addition to water, within our work group, we also review data on microplastics found in aquatic organisms, or biota, as Rebecca said. And this figure brings together results from 9 different studies that examine 28 different species across several groups of organisms, which are shown in the legend on the right-hand side of the photo. And across the region, microplastics have been consistently detected in biota. Fish are the most commonly sampled group, and interestingly, they are the only group that has been studied across all 5 Great Lakes. And from a human health perspective, there have actually been 2 studies that have gone a step further by analyzing not just the internal organisms of the fish, but also the edible portion, or the fillet. So this gives us actually direct insight into potential exposure through fish consumption. Next slide, please. The first of these studies was published in 2021 by McIlrath and colleagues, and they examined sport fish from Lake Simcoe. And then the second was published in 2024 by Milne et al., and that study focused on sport fish from Humber Bay in Lake Ontario, very near to Toronto, where I'm situated today. And both of the studies detected microplastics in the edible portion of the fish. On average, Lake Simcoe fillets contained about 8 microplastics per fish, while the Humber Bay fillets contained about 56. Each study also examined the potential annual intake based on average adult eating fish twice per week. And the more recent study by Milne et al. Calculated that this rate of consumption for Humber Bay sportfish specifically could result in an average intake of about 14,000 microplastic particles per year. So together, these findings on fish, along with the tap water results, highlight that while data on human exposure through Great Lakes resources does remain very limited, the evidence we do have shows that exposure is possible. At the same time, there is a clear need for more data to understand how these exposures differ across different locations in the Great Lakes, different species, and different seasons. So with that, I'll turn things over to Dr. Karen Kidd, and she will discuss the sources and transport pathways of microplastics in the Great Lakes.
Thanks, Eden. So last week you heard Chelsea Rockman talk about the overall production of plastics and how over half of the plastics we produce and used are disposed of. And many of those plastics become trash in our environment. So those larger plastics like straws, bags, plastic bottles, foam packaging, cigarette butts, etc., food wrappers, those macroplastics are eventually broken down into smaller and smaller plastic bits. That's where microplastics can come from, and that's any plastics that are smaller than 5 millimeters in size. So we have that source. This figure here is describing the general sources of microplastics to the Great Lakes and their tributaries, and there's lots of different ones. So for example, in the top left, we know that when we wash our synthetic clothing, there's a lot of plastic fibers that get washed into our wastewater systems and can get discharged along with industrial emissions into our rivers and lakes through wastewater treatment plants. We also know that the biosolids from wastewater treatment plants can be applied to agricultural lands, and that in combination with a lot of the plastics that are used in agriculture and food production, they can run off into waters with stormwater flow. We know that roads are a large source of plastics to our waters through the wear of tires as well as the paints that are used on the roads. I mentioned already the breakdown of plastic litter, and then certainly there's a lot of marine debris that can be a source of microplastics to Great Lakes, so fishing line and nets, for example. And all of these sources of plastics, the smaller particles can get ingested by fish and other aquatic life and cause harm. So the microplastics move through the environment into the Great Lakes with stormwater runoff from urban and agricultural lands in wastewater effluents. They come into the Great Lakes from the tributaries, and they're also carried through wind and in rainfall. Next slide, please. So the second objective of our work group was to develop a monitoring framework for the Great Lakes. And that's because while there's strong evidence, as you just saw from Rebecca and Eden's slides, to suggest that microplastics are present throughout the whole Great Lakes Basin, much of this evidence is based on peer-reviewed literature that's often very geographically limited in its scope. You know, it's done over a short time period, and a lot of these studies are not done using harmonized methods. So the Great Lakes Basin would really benefit from a routine basin-wide monitoring program that would really help us identify whether microplastics levels are getting better or worse over time and identify where the hotspots are. Do we see more microplastics in tributary waters versus lakes, more in sediments versus beaches, for example, and how much is in biota and what kind of risks does that pose to the biota and their consumers? We really need to make sure that we're comparing apples to apples across the Great Lakes Basin. So what we did in September of 2023 is we had an expert workshop, group. We brought an expert group together. Is it Friday yet? To develop recommendations on standard operating procedures, monitoring methods for the Great Lakes that really will allow us to assess the trends in microplastics in the Great Lakes over time and find those hotspots of high microplastics. So what we did at the workshop is we came up with harmonized sampling protocols, specifically for lake water, river water, for sediments, and for organisms like fish. And the program is designed to capture data on microplastics, both over space and time, as well as where they're coming from and where they're going. And we really are hoping that this information will get incorporated into basin-wide monitoring programs. So the next slide is showing you some of the standard operating procedures that we've published that are available through this QR code here. So there's SOPs or standard operating procedures for lake water, river water, sediments or mud that's at the bottom of the lakes and rivers, and then as well as for organisms. And a lot of these documents lean very heavily on the work that was done in California by SQUIRP that Rebecca mentioned earlier. So the goal of these SOPs is not only to provide harmonized monitoring approaches across the Great Lakes, but to harmonize Great Lakes monitoring with similar efforts in other parts of North America, so including California and Chesapeake Bay. So we adapted these SOPs from the work that SQUIRP had done, but we've also added addendums specific to the Great Lakes Basin. And so the Great Lakes tributary sampling SOP, for example, was written based on feedback at the workshop. We didn't develop one for beaches because They were determined to be less relevant to the kinds of ecological risk assessments and management frameworks we were doing. So Eden will talk about those frameworks next. Over to you, Eden.
Thanks, Karen. So yes, next I'd like to shift to our work group's efforts on microplastics risk assessment and management. But before I dive into the details of our third objective on risk, I want to briefly situate this work within the context of the Great Lakes Water Quality Agreement. So one of our key recommendations, which Karen will share more on at the end of the presentation, is to formally include microplastics in the agreement. And as Rebecca mentioned at the top of our presentation, this agreement represents a binational commitment between the governments of the United States and Canada to protect, restore, and enhance water quality in the Great Lakes. And it's one of the strongest tools that we have for coordinating joint action and shared resources to tackle water quality challenges and safeguard the lakes for the millions of people who live and work and play within the region. So the agreement outlines 9 overall objectives for Great Lakes water quality, and Objective 4 specifically, the one which we're showing here on the slide, focuses on ensuring that the lakes are free from pollutants that could harm people, wildlife, or other living things, whether through direct contact or through the food chain. Next slide. So progress towards this objective is tracked through indicators, which are essentially measurable signals of ecosystem health. So for Objective 4, the main indicator is the presence of toxic chemicals in the Great Lakes ecosystem, which are monitored and reported in the State of the Great Lakes report. So these are reports that are published every 3 years. And the table on this slide shows how these indicators are organized. So along the top, you can see each of the 5 Great Lakes. And then along the left-hand side, you can see the environmental areas where the chemicals are measured. So there's sediment, water, fish, herring gull eggs, and then the atmosphere. And each combination of lake and area receives 2 ratings. The first is a status rating, so either poor, fair, or good, which each has its own color, red, yellow, and green. And then they also receive a trend rating, whether things are deteriorating, whether they are unchanging, or whether they are improving. And governments determine the status by comparing measured concentrations of these toxic chemicals to thresholds of concern, which are basically levels above which harm is to be expected. And then trends are based on how those concentrations change over about a decade. So this reporting structure recognizes that many of these chemicals remain in the environment for a long time. They are persistent. So I'll ask you just to keep those status and trend ratings in mind for now, and we'll come back to them shortly. Next slide. So returning to our work group's focus on microplastics risk assessment and management, our final objective was to develop a framework to do these 2 things, to assess and to manage the ecological risks of microplastics in the Great Lakes. And in January 2024, the Science Advisory Board hosted a workshop to develop recommendations in 2 areas that relate to this. So one, recommendations for an evidence-based framework that can help us understand how different aquatic species might be affected by microplastics exposure and at what levels of exposure management actions might be needed. So this gets back to those thresholds of concern. And then the second is ways to integrate monitoring and effects data into that framework to make it more useful for decision makers. So what are the parameters by which we use this scientific data to inform management options? And the ultimate goal of this was to help interpret the monitoring data, which of course hopefully would be gathered by these standardized or harmonized SOPs, which Karen just spoke to, and support the eventual inclusion of microplastics as one of these toxic chemical indicators that's used in the reporting system that I just mentioned. And as I noted a moment ago, determining a chemical's status under the agreement requires having this known safe level or threshold, so the point at which we know that harm begins to occur. So one of the first steps our group took was to develop these thresholds for microplastics in the Great Lakes. So next slide. And to do this, we use something called species sensitivity distribution curves, or SSDs. So working with our workshop participants, our work group created two SSDs, one for exposure to microplastics in surface water and one for exposure to microplastics in sediment. So shown here is the SSD curve for surface water. And essentially, SSDs are statistical tools that show how different species respond to a chemical or pollutant. So this is created all using laboratory-based data. So tests that expose different species to microplastics in a laboratory. So some species are more sensitive and they appear towards the bottom end of the curve at the bottom left-hand side there, while others are less sensitive and they appear towards the top of the curve. So you end up with this curve that shows how different species are either more or less sensitive to microplastics compared to other species. And these curves are widely used by governments and researchers to help set safe environmental limits across different contaminants, different pollutants, essentially to find the level of a chemical that are expected to protect most species in an ecosystem. So one important value that's derived from these curves is something called the HC5 value, or the hazard concentration for 5% of species, which is shown in orange on this SSD. You can see this little orange half square at the bottom of the SSD. And this represents the concentration at which 5% of species are predicted to start showing harmful effects, or if you flip it, the point at which 95% of species in a community are predicted to be protected from that contaminant. So the SSD that we made here, it's based on microplastic volumes specifically, because the studies suggest that the amount of plastic ingested, rather than simply the number of particles ingested, is a predictor of toxicity. And the effect that we're talking about here specifically is called food dilution, which essentially means that microplastics take up space in an animal's gut, which leaves less room for real food and reduces energy intake and has knock-on toxic effects. And then all of the species that we included in this SSD, I believe there are 16 species in total, they are all considered biologically relevant to the Great Lakes ecosystem. Next slide. So if you recall back from my introduction using the Great Lakes Water Quality Agreement and the reporting structure, each chemical that's reported on is assigned one of 3 status tiers, so good, fair, or poor, which are shown on the right-hand side of this slide. And this means that we need 2 thresholds to separate these 3 categories if we're going to use this in practice. So together with the workshop participants, our group decided that the lower threshold between good and fair, the green and the yellow, should be representative of the HC5 value, which I spoke of before. So this is the one that is predicted to predict 95% of species, or protect 95% of species. And then the upper threshold, threshold 2, which is between fair and poor, we decided that that should be representative of the HC30 value. So this is the value which protects 70% of species. So using the SSD, we could actually determine the numerical value of these thresholds, which are kind of what the orange and the red lines on the graph are showing. So you look kind of on the y-axis, which is the percent of species affected. You draw a line over to the curve and then draw a line down, and you actually get a concentration of microplastics. So for our first threshold, the HC5 value based on our SSD, that was set at 331 particles per liter. And the HC30 value was set at just around 21,000 particles per liter. Next slide. So what does it actually look like to use these values in practice? So that is what we're showing on this slide here. So during the same workshop, we conducted a preliminary comparison of these proposed thresholds with real-world data on microplastic concentrations in the Great Lakes. And this is what this figure is showing. This is for exposure in surface water. So it compares these threshold values, which I just spoke about, to measured concentrations of microplastics across the lakes. So each of the lines shows how concentrations are distributed for one of each of the 5 Great Lakes, and then the 2 thresholds you can see are marked on the plot for reference. And so what our analysis shows is that Lakes Michigan and Lakes Ontario appear to exceed the higher threshold, so that HC30 value, where 30% of species are predicted to be impacted, while the other three lakes, Superior, Huron, and Erie, are approaching or exceeding the lower threshold, so that value at which 95% of species are predicted to be impacted. And of course, this is preliminary because much of this data was collected using non-harmonized methods. So this, we call a preliminary comparison, but we hope within time our harmonized SOPs are used to collect a better data set where we can do a more robust analysis. And at the time of our workshop, unfortunately, there wasn't enough data yet to do this exercise with sediment. So again, that's another area for future work. So with that, I'll turn things back over to Karen, who is going to walk us through the key recommendations from our report.
Thanks, Eden. So as Eden just mentioned, we came up with 4 key recommendations from this exercise. The first of which is to develop a regionally coordinated monitoring program with harmonized methods for the reasons that we just described. And that would allow consistent reporting. It would allow more rigorous ecological risk assessment, an understanding of whether mitigation methods were successful, and overall better management of microplastics in the Great Lakes. So that's the first one. Our second recommendation is around research to refine monitoring and analytical capabilities and ecological risk assessments. So this includes support for developing better lab methods to characterize Great Lakes microplastics, improve their detection, and increase throughput in analyses. The continued research should also include testing monitoring techniques and expanding our understanding of the ecological effects of microplastic exposure on aquatic species, especially those relevant to the Great Lakes and especially for sediment exposures. As you just heard, we don't have a lot of data on how microplastics and sediments affect aquatic species. The third recommendation is to help, to coordinate monitoring, assessment, and management actions. Microplastics should be evaluated under the Great Lakes Water Quality Agreement as a toxic chemical sub-indicator and as a chemical of mutual concern. So that was our third recommendation. And then the last one is certainly that we need to take steps to prevent the emissions of plastics and reduce plastic pollution in the Great Lakes, which will reduce environmental levels and prevent further ecological risk. Next slide, please, Rebecca. So since we released this report to the commissioners in November, and we had a public webinar on it in February of this year, there's been a lot of uptake and interest in the results, including this webinar. So thank you very much to the foundation for this opportunity. But here's some of the impact that has come out of our recommendations. So the first is that the Great Lakes St. Lawrence Cities Initiative passed a resolution endorsing our recommendations. The next one is the Great Lakes St. Lawrence Legislative Caucus adopted a resolution to encourage the adoption of microplastics as a chemical of mutual concern. The third is the Michigan Microplastics Coalition sent a letter to the Great Lakes Executive Committee supporting the SAB's recommendations. Next slide, please. And a few more to mention is that the state of Michigan legislature introduced several bills that cite this SAB report. So I'll leave those to your reading here, the 2 bills, but the first one proposes a statewide microplastics research and monitoring plan. And then the second one proposes amendments to the Safe Drinking Water Act to address microplastics contamination. And then the last impact we wanted to mention was at the recent Great Lakes Commission annual meeting, there was a resolution to develop an action plan on microplastics. And there was a call for coordinated basin-wide efforts to reduce plastic pollution through monitoring and assessment. So you can access that through the PDF there, the link to the PDF. Next slide. So in terms of what you can do around the issue of plastics and microplastics pollution, certainly you can find your MPP and MP and write letters encouraging the reduction of single-use plastics, for example. You can check out the U of T trash team that Chelsea Rockman talked about last week. There's a lot of great tips on how to reduce your plastic waste, and there's a screenshot here of some of them. Things like avoiding single-use plastics for drinking, bringing your own water bottle, coffee cup, for example. So I encourage you to check out their website for lots of great ways to reduce our use of single plastics. Next slide, please. And we've talked about this slide already, so just a lot of people were involved in this work. So we have many people to thank. Many organizations contributed to the outcomes of the work. And then the last slide is, again, the QR code that will take you to the report, as well as to links for the standard operating procedures, as well as the many appendices from our workshops. So thank you very much for your listening, and we're happy to take any questions.
Thanks so much, Rebecca, Eden, and Karen, for such an engaging and insightful presentation on the report, its findings. I know I learned a lot, but thank you for sharing all of that. With that, we have time for questions and discussion, so I'll open it up. We have a question in the chat from Gail. Gail, I can unmute you if you'd like to ask your question.
Hello, everyone, and thank you for the great presentation. I was curious, when you say that you test how well an organism can survive in the lab, and how representative that would be in a lake environment when there's all sorts of other things going on, other contaminants in the water. And is it possible to test in situ?
I can speak to that a bit. There's two things that come to my mind. That's a really good question, and I think it's a challenge the field is grappling with, but has made improvements on in kind of the last decade or more of microplastics research. So there's two things that come to mind. One is the diversity just of microplastic particles that exists in the real world. So we know there's many different polymers, many different sizes, many different shapes, like really probably every single microplastic particle that is encountered in the environment is different from the next. So when toxicity testing really started, we really used a lot of spheres, perfect spheres for shape, and there maybe one size and one polymer. So you really didn't capture the diversity of microplastic particles in these toxicity tests. So yes, they probably didn't maybe perfectly replicate what animals were actually exposed to in the environment, but the field has made improvements with that in the past decade or so. Like now we test a lot more different shapes and sizes and polymers, and we test range of these different characteristics in a single test. So you'll get a test with a range of sizes or perhaps shapes or polymers, but the field is still working on kind of maybe a standard mixture of microplastic particles that really represents what's found in the environment that can be used in a laboratory test. So although we've made improvements, there's still further to go, and there are a lot of researchers actively working on this to try to get a very realistic scenario in the lab. And then the second part of your question that I heard was kind of the interaction between microplastics and other contaminants that exist in the environment. And there are studies that do this as well. So they'll vary different parameters, whether it's temperature, salinity, pH, while exposing organisms to microplastics, or they will use microplastics with a co-contaminant. So whether that is PAH or a different type of POP, another persistent organic pollutant, and they'll do kind of co-exposures. Our SSD, though, only includes microplastic particles. So it doesn't include any of those studies that also look at co-exposures with other contaminants other than microplastics. Yes, but that is it's a good question, and it's a challenge that the field is definitely grappling with, but we've made improvements for sure.
And when you say that you might take a range of polymers and or a range of sizes, would that vary from one lake to another? Like, would it be different in Lake Ontario than what you would try and test in Lake Superior or Lake of the Woods or Rainy Lake?
Yeah, it very well could, because that will be linked to the sources to those specific areas, which can be localized depending on the activity that occurs within the watershed of those lakes. So for sure, if you wanted to get really realistic, you could do an assessment based on a localized region and then match, what you find in the lake to then what you use in the lab. Yeah. So we'll see what comes out of kind of the standardized work that's happening in the world. I don't know if they're going so specific, thinking about what each different region in the world would look like, or if it's more just kind of a generalized mixture of microplastics that kind of represents what you find in the environment globally.
And I'll just add a couple of things in there. One of the really exciting projects that's going on is that whole lake experiment that Chelsea Rockman presented last week. And that's going to really help us understand what it means for microplastics to be in lakes. Like, how does it affect populations of fish and the food web? So I'm really excited. I know there's lots of people that are very excited to see the results of that work, and Rebecca's involved, and Eden they're both involved in that experiment. And we also are getting a better sense of what fish and other species are getting exposed to. So there's more studies recently on the types of microplastics that are getting into the guts of organisms and into the tissues. So I think that information will be really helpful to link to the lab exposures and really get a better sense of the risk of these exposures to aquatic species. Thank you.
Thanks, Gail, and thanks for everyone answering the question. I'll move to Bree. I'll unmute you.
Hello? Megan?
Yes, hi, we can hear you.
Okay, it's Brady.
Perfect. Hi, Brady.
Hi. Karen, you kind of made me laugh there when you just answered that last question about being excited. And I am more scared than being excited, to tell you the truth. And great presentation by you girls. This is really good information. I'm more concerned, like right now, if you personally, where do you get your water from? Like, do you use drink out of the tap, or do you have water filters on your tap? Do you drink bottled water? What, like, this is concerning, like very concerning to me, as to what it should be added maybe to the list of what we can do at home as to drinking, making sure that our water that we're consuming is clean. And I'm curious, is there a filter that you could possibly buy that you could recommend?
I don't know that we could endorse any sort of particular brand of filter, but I would say there's evidence that microplastics leach from bottled water into the water in the bottles, especially if they're like left in the sun. So I don't really drink bottled water. I drink water from the tap. And if you are concerned about microplastics in your drinking water, I think, there are other contaminants that could also be filtered out by filtering. So something like a simple Brita filter is a good way to provide extra polishing to your tap water. I don't know if Karen has additional information about drinking water, but we very carefully in our scoping of our microplastics work group focused on the impacts on environmental health and stayed away from impacts on human health because at the time there was just less known about the risks to humans from consumption and the potential health impacts. I think more is coming to light even in just the last sort of 3 or 4 years since we started this work group. And I hope there will be more synthesis about that research forthcoming, but our report doesn't really speak to the human health risks.
Yeah, I think it's the unknown that I'm more concerned about. So, like if there's something that we should be doing, like I'm not sure if the Brita will get rid of microplastics or not. Maybe it does. I don't know, but it might be something that some of these companies that make these filters, if you do come across one that or a company that produces and promises to help the drinking water, they may be able to fund your work here, and because it's important work that we get to the bottom of what's happening to our water and what's going to happen to us as to our fish and to our wildlife. And if it ends up well, it definitely will end up in us. So it creates a major problem. So there I'd like to see something be done as soon as possible as to trying to keep the water clean. Like I know that, personally, all the things that you recommend, about cleaning the water and stuff, I know that I can do that. This group can do that. But there's 40 million other people in Canada that aren't getting this message and hearing what you're saying. So this is very important. It's very concerning to me, and it should be to everybody. So, great presentation. I will certainly be putting on some kind of filter on my tap water, just to let you know that. I'll do my research and see what I can come up with.
Thanks, Brady.
Yeah,
Thank you.
thanks, Brady. Thanks for being here too and learning about this important work. It just highlights again how vital all of the work that everyone here is doing. There's a few more questions in the chat and we have a bit more time for it. So I'll go to Samantha Blackwell. I'll unmute you if you'd like to ask your question.
Sure. Yeah. I was just wondering, there's been 2 attempts in Ontario to introduce a bill to mandate microfiber filters for all washing machines sold in Ontario. And they've both kind of stalled at the second reading. And so I'm wondering, what do you think of the potential for this sort of solution? Because textile um pollution is I think 30% or a large portion of the microplastic pollution in our waterways. Mhm.
Eden, is that something you're on top of? I'm not.
Yeah, I know of this bill a bit. An organization called Georgian Bay Forever was really big in Ontario about pushing forward this bill and working with the MPP, who I think put the first one on the table. And I think France is maybe the only other country globally that has actually passed and is implementing something like this. But yeah, I think any sort of policy or program that stops microplastics at the source before they enter the environment is wonderful. And filters on machines to me is low-hanging fruit. I think that is very feasible, manageable. and could make a big difference. We have had some research out of Chelsea Rockman's lab with previous students that have actually looked at some aftermarket filters that you can purchase and put on your machines yourself. They make a big difference. They trap a lot of microfibers. And so this bill, I think, more is about though companies building them into their products before they actually get put on market. So yes, I think any sort of effort that traps things at the source before they get released to the environment is great and needed, because of course, once microplastics get into the environment, it's really hard, maybe impossible, to pull them back out. It's not like these larger plastics, which you have a better chance at removing from the environment. Microplastics, they go in, and we're realizing that sinking and getting buried in the sediment is a very important fate pathway for a lot of these microplastics. And once they're in the sediment, it's, I think even harder, impossible to remove. So I think trapping them before they get released into the environment is a wonderful way to move forward. And washing machines is a great effort to be made, because microfibers are so prevalent in the Great Lakes. I think our report found that they were the most common shape, microplastic shape, across, I believe, all of the different environmental compartments we looked at.
Mhm.
Thank you so much.
Thanks, Samantha, and thanks for answering that. Our next question from Mike. Mike, do you want to ask your question? I'll unmute you here. Okay, if not, I can read it out loud. So from Mike Staten, can you harvest microplastics from the environment to be used in control lab tests on organisms?
So we have done some of that. We did some exposure trials where we collected biofilm from natural environments where we knew the microplastic exposure, and then we fed that to snails in a sort of ecotoxicity test. And I think that there have also been a lot of efforts to do in situ exposures, where you might have like tadpoles or small fish in an enclosure that's then placed into the natural environment to see what effect exposure to kind of a diverse collection of microplastics in the environment is. But I don't know about many studies that have done collecting like a water sample of environmental microplastics and using that. I don't know, Karen, do you have examples of that?
I don't. I think the ones I'm familiar with are like the ones you've mentioned already. Mm-hmm.
There was
again,
one study out of our lab by a PhD graduate named Kennedy Bucci, and she actually did get microplastics from Lake Ontario and then bring them into the lab and do a exposure with fish. I can post the link to the study in the chat. I don't know about the outcome of the study, but I can post the link if anybody wants to follow up and check it out.
Thanks. In addition to the mixture of particle types like sizes, shapes, colors, and additives and polymers that would occur between these kind of like purchased microbead microplastics and what you'd see in a realistic exposure, there's also the weathering. And so microplastics in the natural environment will start to degrade, and their surface can change in terms of its physical shape and roughness, and it can absorb other contaminants and be colonized by bacteria and algae. So it does really change the palatability, the buoyancy, the behavior of the particles to have them be weathered in a natural environment. And I think we'll see more of those sorts of weathered particles used in ecotox experiments, as that's now been pretty well recognized.
Thanks for those answers. Mike just wrote in the chat that his microphone doesn't work, but thanks so much for the reply. We have one more question, and I think that would be it for timing. So Matt, do you want to read your question? I'll unmute you.
Sure. Can you hear me, Meg?
Yes, yeah.
Thanks, and thanks for your solid work on this series. Awesome. And thanks for the presentation there today. I guess my question is surrounded around the fact that I'm aware of some really good work that's being done in this watershed, Rainy Lake of the Woods watershed, to develop a core monitoring program for the watershed. And I noted that, earlier the experts on this call indicated that, most plastics, the areas that have most plastic concentrations are associated with high population centers. And I'm not sure our watershed would be considered a high population area, probably the opposite. And just unsure the degree to which microplastics should be on the radar screen for this watershed, even though they are a concern of myself personally, as well as I know as the community around us. So that leads to the question: are you aware of SOPs, strategic or standard operating procedures available that would provide direction to watersheds? What would be given the amount of resources we have to do this type of monitoring, is there a sampling protocol that provides a minimum sampling for watersheds to understand their baseline condition for microplastics? Is it 1 or 2 per? Is it on a sub watershed basis that this would be done? And yeah, so Chelsea did a great job of describing a standard operating procedure for a sampling protocol last week. That was excellent. Raised my eyebrow. Just wondering how we'd go about doing it with the resources we have in this watershed.
So I think, Matt, you're asking about more like a monitoring plan. So not so much about the details of how you'd collect an individual sample, but how many samples you need and how frequently and how spatially distribute those across the watershed. Is that right?
Absolutely correct.
Yeah. So our report doesn't include guidance on that. We ended up focusing on how individual samples were collected and processed, and some of the minimum reporting requirements to make sure that that's really reproducible. But I think we did have discussions about monitoring planning, and I would say universally everyone is feeling resource trapped. And so there was a lot of concern about, saying, and just tack this on top of what you're already doing, right?
Exactly.
One thing I would say is that for macroplastics, so that bigger litter that Karen was kind of describing, there are publicly available protocols that can be done, say, to do like beach surveys that involve community scientists or citizen scientists, and things like that can be incorporated that maybe would provide some information about the extent of plastic pollution that don't require a lot of expensive lab analyses. And so that might be something to look into, because those, as sort of Karen explained, those macroplastics break down to make the microplastics. And that can give an overall index of like where you might want to target some strategic water or sediment or biota sampling to look at microplastics. What do you think, Karen? What do you think, Eden?
Yeah, I think that beach survey is a great idea. And I also think that some surveys would give you peace of mind. So even if there aren't a lot of people in the basin of Lake of the Woods, at least having some data in hand to compare against these thresholds that we've derived would be, I think, important to do. I understand resources are limited, but I don't think you would need a lot of samples. You could put them strategically in places where there are more people to get a sense of how hot potential hotspots might be, but it wouldn't take a lot of effort, I don't think, to get some baseline data for Lake of the Woods, and then some monitoring over time would give you a sense of whether the microplastics levels are getting better or worse. I'm hoping you can make it work.
I just put in one more resource in the chat too that might be helpful. It's called the Representative Sample Volume Predictions Tool, the RSVP tool. And one of the challenges in microplastic sampling is determining what volume of water you actually need to take in order to reach your my goodness, I'm blanking. The minimum, your limit of detection, to get above your limit of detection. So this tool was created to help people predict how much volume they should take based on what they think the concentration may be, depending on if you're in a really populated area or less populated area. So that's a tool that you could use in your planning of a monitoring program, or just a little study to get a baseline to figure out what volume of water you might actually even need to take.
Thank you very much.
Oh, sorry, Matt. I just put them in the chat. I think before it had just gone to the host and panelists, but everyone should be able to see them now.
Thank you.
Great. And I think for timing, we should wrap up now. But Matt, thank you for asking your question. I think that's very relevant. And if you saw Chelsea's talk last week, she was also talking about community monitoring for microplastics and has some good resources that she shared at the end that might be worthwhile for us to look at as we move forward with thinking about microplastics in our basin. But thank you so much to our presenters, as well as everyone who joined this afternoon and contributed to answering questions. I think this was an awesome discussion, and I'm glad we had so many insightful questions. As a reminder, this session is recorded and it will be available on our website. You can also find the recordings of our previous webinars and register for upcoming ones. Our next one isn't until November 21st at 1 p.m. Central Time, and it will be on updates on aquatic invasive species, specifically zebra mussels and decontamination practices in and around Lake of the Woods. That will be featuring Brendan Spearin from DFO and Chris Herc and Michaela Novak from Grand Council Treaty 3. So I'm looking forward to that. Hopefully you all can join us. Thanks everyone, take care, and we'll see you at our next session.
Thanks very much.
Bye-bye.
Bye.
Bye.